The study of microbiology has brought us closer than ever to enabling us to alter our very DNA, which can help protect us from nasty diseases. The potential is enormous, enough to possibly change our world. Before we discuss more, let's take a quick look at the small world we'll be exploring and learn of the terms we'll use. The most important molecule involved is probably deoxyribonucleic acid, or just DNA for short. This double helix structure stores the information required to build everything our cells need to function. As you can see, it looks very much like a twisted ladder. Each of the rungs or steps of the ladder are made of only 4 complementary bases. The letters stand for adenine, thymine, guanine, and cytosine. Adenine pairs with thymine, and guanine pairs with cytosine. It's these stored sequences that determine how the essential components of the cell are built. Proteins. Proteins are made up of smaller molecules called amino acids. When DNA is transcribed into mRNA, a single-stranded molecule which matches the DNA sequences it copied, the mRNA carries the code needed to assemble the amino acid chains. The chain is built inside a big factory of sorts, called a ribosome. Once this process, known as translation, is complete, the ribosome releases the newly formed chain to be folded up into a protein. Now, these proteins can perform their specific jobs which allow the cell to work. The cell can do many jobs in a multicellular organism. We ourselves contain around 220 types of cells, all doing different things in our body, but many trillion actually make up the body itself. All cells have one thing in common, though, and that is replicating themselves. This process is essential for any cell, but also one that can cause serious diseases. Cancer is a disease caused by mutations or mistakes in the replicating machinery, which can lead to uncontrolled replicating. We've tried using radiation and other techniques to stop this biological disaster, but there might be another way to use custom DNA sequences to destroy cancer before it becomes a serious problem. DNA sequences can be sought out using special machines such as proteins or restriction enzymes, which can take out vital DNA required for cellular processes. If, for example, part of the DNA used for replicating is taken out, the cell won't be able to replicate itself without it. This idea could be used to stop cancerous cells, as long as the DNA used to code for the cutting machines can get into the cell. But is there a way to get this special DNA into a cell? There actually is, and one way is to use viruses. Viruses are little more than a small sphere of proteins filled with RNA and sometimes DNA. They attach to cells and inject their nucleic acid, which is then transcribed and translated into proteins it coded for, while using the cell's own machinery to do so. This continues on until the completed viral replicates exit the cell, which may include destroying the host cell in the process. The same technique could be used to deliver our custom DNA sequences to a cancerous cell, replicate our virus while producing the cutter protein, and disable or destroy the cell in a controlled manner using the cutters. This new kind of technique is currently being investigated by scientists, but is still in its early stages. The CRISPR-Cas9 system is an example. I won't go into its detailed processes, but it can basically seek out sequences in DNA and either cut it out or replace that sequence with a new one. Using proteins and systems already employed by cells is a fascinating way to edit DNA. Developing these new technologies might bring some deadly diseases to an end, and also open up a whole new world of DNA editing. Hope you enjoyed. Thanks for watching.